In this module of biotechnology, let us talk about use of selectable markers and insertional inactivation. Selectable markers are some artificial gene sequences that we introduce inside a vector which will help us to develop some kind of phenotypical expression that we can see or measure from outside. When we talk about vectors, our vector should have selectable markers, and we took two examples for the selectable markers. One was an antibiotic resistance gene and another one is enzyme producing gene. Now let us see how we use these markers to choose between transformants and non-transformants. What exactly are transformants? Transformants are cells that in which the new gene or foreign gene or desirable gene has been introduced. Say we are talking about ampicillin resistance gene. We know the vector PBR322 has ampicillin resistant gene. Suppose this is our plasmid and in this we have introduced our desired gene. So this is our insert and this is our plasmid. In this plasmid PBR, here is this ampicillin resistant gene, and if we introduce this rDNA into the host, we want to check whether the rDNA has gone in or not. Say these are our E. coli cells and we introduce this rDNA into this E. coli. This E. coli received the rDNA, this also received, but these three did not receive. This is what exactly we want to check. If we culture this E. coli, 50% of E. coli are not going to produce anything for us because the insert has not gone in. Only 50 will produce. So to get rid of the non-transformers, we will grow them in a culture medium containing ampicillin, because we know that if our rDNA has gone into it, along with it ampicillin resistance gene also has gone in. By this, the E. coli where the rDNA could not penetrate will die. So, using this ampicillin resistant gene as marker, we have selected our transformants and eliminated non-transformants. Second method is by using an enzyme producing gene. The gene is most commonly used gene is XIZ gene, and the enzyme produced by it is beta galactosidase. To understand what it does is, a substance called X-gal, which is a modified version of galactose, which is white in color, is converted into a product which is blue in color with the help of beta galactosidase. This is what we are going to use. So this is a plasmid which we are going to use as a vector, and here is our LAC-Z gene, and in this case we introduce the insert in this gene. This red color is the insert. By doing this, we have inactivated this gene which is responsible for the conversion of X-gal into blue product, and that is why this process is also called as insertional inactivation. And how are we going to use this? Again, same thing. These are E. coli which we want to test transformed or not. Actually, by this insert, LAC-Z gene is inactivated and beta galactosidase will not be produced. So, the white color X-gal will not converted into a blue color product. So, the white color colonies of E. coli are transformants and blue color non-transformants. Insertional inactivation is also called as blue-white conversion. Hope now the use of selectable markers and the process insertional inactivation is clear. Thank you.